Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to be talking about ganglionic synaptic transmission in the autonomic nervous system. So with that, let's begin. So I'd like to start off by describing to you how neurotransmitters are going to be released from the preganglionic neurons. And this is going to occur through the following process. So inside the preganglionic neuron, you have vesicles stored at the active zone filled with neurotransmitter. In this case, it's going to be acetylcholine. So what happens is, in order to release the contents of these vesicles, you need to have a signal. And the signal is going to start off with an action potential. So action potentials propagate down the axon, opening voltage-gated sodium channels. This allows sodium to flow into the cell and therefore depolarize it. So this depolarization will then open voltage-gated calcium channels, which allow calcium to flow into the nerve terminal, increasing the intracellular calcium level, which will then facilitate the process of vesicular fusion, which will allow the contents inside the vesicle to be released into the synapse. So now that we know how acetylcholine is going to be released by the preganglionic neurons, we're now going to talk about a specific concept before we get into what acetylcholine does when it binds to the receptors on the post-ganglionic neurons. So let's take a look at this concept first. So the concept that I'm referring to is convergence and divergence in ganglia. So what is convergence in ganglia? So imagine you have a post-ganglionic neuron right here in blue, and this post-ganglionic neuron is actually going to be interacting with a number of preganglionic neurons. We have a red preganglionic neuron, a purple, and a yellow. And the common theme with all of these neurons here is that they're all projecting onto the same post-ganglionic neuron, the blue neuron. So in other words, the blue neuron is receiving signals from the yellow, the purple, and the red. And if all of these neurons release EPSPs, or excitatory postsynaptic potentials, which are produced from acetylcholine binding to a specific receptor, these EPSPs can all add together, and if they reach the threshold, it can produce an action potential in the postganglionic neuron. So convergence is what allows postganglionic neurons to fire action potentials if any individual EPSP is too weak. Because if you have a lot of EPSPs added together, this will allow the cell to possibly reach the threshold and fire an action potential. So now we're going to talk about divergence, which is the opposite. So in divergence, we have a preganglionic neuron that is synapsing onto multiple postganglionic neurons. And in this case, it's going to send signals into all three. So now let's talk about what these EPSPs are. And these EPSPs, or excitatory postsynaptic potentials, are going to occur in the postganglionic neurons. And we're specifically going to be talking about fast EPSPs. So let's take a look. So on the postganglionic neuron, on the cell membrane, you have the nicotinic receptor. And the nicotinic receptor has a reversal potential of 0 millivolts. So when acetylcholine is released from the preganglionic neuron into the synapse, it can bind to the nicotinic receptor, causing it to open. Now, the inside of the postganglionic neuron is negatively charged, and this allows cations to flow into the cell. This produces a current that carries positive charge, and this positive charge is going to depolarize the cell. This depolarization that's produced can be modeled on this graph here, where we see that the cell starts off at rest, it depolarizes slightly, and then it comes down to the resting potential. So this process here is called a fast excitatory postsynaptic potential, or fast EPSP. Now, if you were to look at a postganglionic neuron on the time scale of seconds, and you were to look at the membrane potential that is produced from this time scale, what you would see is something like this. So this is after we stimulated a preganglionic neuron, and this preganglionic neuron released acetylcholine onto the postganglionic neuron. And this right here is modeling the membrane potential of the postganglionic neuron. And what we see in this graph here is two responses. The first is the fast EPSP, which is produced by acetylcholine binding to the nicotinic receptors. But the other thing that we see here is a slight depolarization that lasts for the time scale of seconds. This is a slow EPSP. And slow EPSPs are found in both the PSNS, or the parasympathetic nervous system, and the sympathetic nervous system. And these slow EPSPs are going to be formed from the activation of muscarinic receptors, or GPCRs, on the postganglionic cells. These muscarinic receptors can produce both slow EPSPs or inhibitory postsynaptic potentials, or IPSPs. And these signals, these slow signals, act to basically modulate the postganglionic neurons and modulate the signal further. So in order to understand the importance of these muscarinic receptors, let's talk about a specific example which is related to something called the M current. So what is the M current? So the M current is a current that is produced by a potassium channel. This potassium channel is stimulated by a slight depolarization which opens it, allowing potassium to flow out of the cell. And this produces a slight hyperpolarization inside the cell. So what is the effect of this hyperpolarization? Well, the effect of this hyperpolarization can be modeled with this computer here. So what we're going to do is we're going to put in a small stimulus and we're going to sustain it for a while. And what we're going to do is we're going to measure the response in the postganglionic neuron. And what we see is something like this. So the first thing that we see here is that we have an action potential that is produced in the postganglionic neuron. But then what we see here is that even though the neuron is slightly depolarized here, the neuron does not produce an action potential. And the reason why is because of the slight hyperpolarization caused by the M current, which is from this potassium channel. The M current slightly depolarizes the cell, therefore preventing another action potential from occurring. You only get one spike. So now, what if we were to bring in our muscarinic signaling? So if we were to inhibit the M current by adding muscarine to the synapse, the muscarine would bind to the muscarinic receptors on the postganglionic cell, and the activation of these receptors would inhibit this channel. So when we repeat the experiment, we would get something like this. So what we see here is that by inhibiting the potassium channel, or by inhibiting the M current, we can do this by activating the muscarinic receptor. So when we activate the muscarinic receptor, we inhibit this potassium channel, which causes the cell to lose the influence of the M current. This therefore causes an increase in the excitability of the neuron, which makes it easier to fire action potentials, which is why we get this lineage of action potentials firing. So this shows you how important the muscarinic receptors are in modulating signals for the postganglionic cells. So now what we're going to do is we're going to talk about how acetylcholine is recycled. Because remember, we had acetylcholine that is released from the pre-ganglionic cell onto the postganglionic cell. But in order to stop the signaling, we have to either remove the acetylcholine, remove nicotinic receptors, degrade these receptors, or we can basically degrade acetylcholine. And that's what we're going to be talking about here. So this is the preganglionic nerve terminal, and this is the postganglionic cell. And what we're going to do here is we're going to first see how acetylcholine is formed. So inside the preganglionic nerve terminal, we have mitochondria. And the mitochondria is going to produce choline and acetyl-coenzyme A. Now, choline and acetyl-coenzyme A can be reacted together by using the catalyst choline acetyltransferase, which catalyzes this reaction. So the reaction that this enzyme catalyzes forms acetylcholine. Now, in order to get acetylcholine into the vesicle, you need to have a transporter. And this transporter exchanges acetylcholine for protons. So what also happens is that the protons are going to be recycled back into the vesicle, and at the same time we also have ATP being pumped into the vesicle as well. Now, in order to do this, you have to have calcium, and the calcium is going to come in through voltage-gated calcium channels, which allows vesicular fusion to take place. And when the vesicle fuses, this releases acetylcholine into the synapse. Now, acetylcholine at this point can bind to nicotinic receptors, but in order to stop the signal, you have to remove acetylcholine, which is one of the steps. So acetylcholine can be degraded by acetylcholine esterase, an enzyme. So acetylcholine esterase hydrolyzes acetylcholine into acetate and choline. Then acetate and choline are transported back into the nerve terminal, which allows the cycle to take place all over again. So that's it for this video. I hope it helped you understand the ganglionic signaling that occurs between post and preganglionic neurons, and I hope to see you in the next video. Thank you for watching, and I hope to see you next time.